So I'm assuming you have completed the notes related to Lab S4. Now let's continue with the lecture. So this slide shows you different types of neurons with their dendrites. You'll notice, why do neurons have such an elaborate structure of dendrites? So these are dendrites, by the way. This is a cerebellar Purkinje cell. This is a spinal cord motor neuron. This is a neostriatal spiny neuron. And this is a locust interneuron. This is from the insect, locust. Why all these fascinating and elaborate structures? What function do they serve? What is it for? Okay. So it is not very clear, although we can have good guesses. Probably it's an antenna. These are antennas to receive inputs, meaning spikes from other neurons. So other neurons convey their input via synapses on all of these elaborate structures. A neuron is thought to receive, in a human brain, is thought to receive inputs, thought to have approximately 10,000 to 20,000 synapses, meaning in a neuron about the size 500 micrometer, there'll be 20,000 synapses possible. Okay. Where is the soma in this case? In some, it is very difficult to even identify where the soma is because it's full of dendrites. And soma performs the function of integrating, receiving. So all of this elaborate structure perhaps serves to receive inputs, integrate them, meaning decide what to do with them. If overall excitation is greater than inhibition, that's the meaning of integration. Cell may decide to fire. That's typically what happens in cells. So these synapses will be excitatory and inhibitory. If overall at any instant the excitation exceeds inhibition, it's possible the cell may fire. That's captured in this word integration. Okay. And then once it decides what to do, it'll communicate. So if there is a spike, it'll communicate the spike via axons. If there is no spike, it doesn't do anything. It's silent. What part of the neuron does what? What is the division of labor? Okay. So we need to consider multi-compartmental models of neurons. So far, we've considered only one or two compartments. A neuron like this is called a pyramidal neuron, has many compartments. So a soma would be here. These are apical dendrites. Remember, we are trying to capture these in models. So this is obviously a reduced order model. So instead of all these elaborate dendrites, we have three compartments here. That soma is the soma. And these are basal dendrites, just two instead of so many here. And then this is the axon, line with an arrow. That would be the axon, the output. So this is called a reduced order or a stylized model, simple model of the pyramidal cell. You could develop more complicated models like this. This is for a Purkinje cell that has 4,550 compartments, developed in 1994. It has 8,021 channels. So you can develop fairly detailed models. These days, there are models. We have a model in our lab, which has over 1,000 compartments and over 20,000 synapses. Okay. So going back, these are called multi-compartmental models, ranging from simple ones to very, very complicated ones. These are called very realistic models. Okay. Again, computational models so far have been single compartments or two compartments, which you saw in S3. You recall. So the basic neuron types from biology are there is a bipolar neuron, unipolar neuron, multipolar neuron, something like pyramidal cells, different types. Okay? So the question we are asking is, can a biological neuron be modeled as a single compartment as we did in lab S2? So that depends on the application. If you want to worry about just the spikes, how a neuron spikes, a single compartment is good enough. And for theoretical studies, people use single compartment too sometimes. Multi-compartmental model of a neuron would consist of dendrite compartments, soma compartments, and axon compartments. And then you take these models of single neurons, and then you make networks of multiple cells. And then models of brain regions themselves. And then the idea is that we could have a model for rodent brain. There are groups around the world working on that. Now, if you have all these neurons, and it's very clear you want to make them talk to each other, we have to understand how do they talk to each other. Otherwise, you cannot develop these models of networks of neurons. Like we motivated you, probably the role of the dendrites is to receive. Integration role of integration probably is soma and the initial segment of the axon. They serve that job. And what about communication? Communication. So this communication is via synapses. And you must have seen from biology, all the videos and all that, how it works. I'm going to be very, very brief about it. This is, again, a very interesting, a stylized model showing dendrites, nucleus, cell body, axon. This is axon, which may or may not have myelin sheets. And then these axons go and end in synaptic terminals. So if you look at this, zoom in at this, that's what you see here. You see this presynaptic cell coming in from this axon. And then this is a synapse. So this blue one is for the postsynaptic cell. Assume there is a dendrite here or the postsynaptic cell. So this is a part of that dendrite. So a synapse is a structure that's got two components. The presynaptic component is on the presynaptic cell. The postsynaptic component is on the postsynaptic cell. So there is a cleft in between. So the way a neuron communicates is when this voltage high, the spike comes down. Spike is nothing but an increased voltage, right? When the spike comes down, calcium channels open and they help release on this presynaptic side, and they help release vesicles, neurotransmitter vesicles, and that are docked in the presynaptic terminal. And these vesicles come and fuse with this membrane and then release neurotransmitters. And these neurotransmitters go across this cleft. This cleft is about 100 microns or less. It may vary. So these neurotransmitter goes and binds to the postsynaptic receptors, and then that binding causes them to open. So these channels are called neurotransmitter-gated channels. These do not work on voltage. They work on binding of neurotransmitter. So if that neurotransmitter like AMPA, GABA, or NMDA is the channels, these channels, that let's take AMPA channel. In AMPA channel, the neurotransmitter used is glutamate. So if glutamate is released from these vesicles, glutamate will go and bind on this AMPA receptor, will open this AMPA receptor, will allow a flow of current. Remember, there was a spike that came down, which was electrical. It's converted to a chemical transmission here, which is again back to electrical on the postsynaptic side. Again, please view those videos and try to understand the biology well. So this provides you with more details of what I just mentioned. So these glutamate, in the case of AMPA, has to go and bind on with this receptor at certain points, and then it'll open it up and let it go through. Okay. Now, if you understood the biology, you understood the hardware, the question then will be, how do we model it? Again, showing you the structure, the hardware. The way we model it is, understand it, is try to see that's where theory laws come in. Well, this should be similar to the leak sodium channel. This is again another channel. It's a protein pore. It's a receptor. These are all, by the way, called receptors also. Protein pores. This is also a receptor, a protein pore. Okay, well, that's good to know. So we know how to model this. Do you remember how to model this? Leak channel? Can you write out the equation? Think about it. Can you write out the equation for the leak channel? For the leak channel, it was I leak is equal to G leak times V minus E leak. What about sodium channel? It's INA is equal to GNA times V minus ENA. Standard. And GNA had M and H and all that. KDR, IKDR is equal to GKDR times V minus EKDR. What I've been talking about right now is Ohm's law, essentially. What about calcium? ICA is equal to GCA times V minus ECA. Just make sure you understand how Ohm's law is what governs the functioning of these, if it's ionic flow. So if you think about it, that same law should govern this, Ohm's law. So there has to be some I sin is equal to G sin times V minus E sin. And that's exactly right if you have thought about that. They should all have similar equations and all governed by Ohm's law. Think about it. Otherwise, pause this video, view it again. Now I've given you the answer for this already. What is the equation for GNA? I'll let you fill it. If you're not clear, go back to slide S2 and understand it. How is GSyn different? I'd like you to understand this. How is GSyn different? Any thoughts? Can you write it down quickly? I'll give you just a few seconds here, or pause this video. It's extremely important, remember, that you do it when I ask you to, because that thinking will help you understand when I give you the answer. So please pause the video and write this out. Okay, pause it. I'm assuming that you have paused, so let me proceed. What is the equation for GNA? GNA is M cube H times M cube H is times G bar Na. G bar Na is the maximal conductance. M cube H is how much, what fraction of the channels are open. If M cube H is 1, all the channels will be open. So the conductance will be G bar Na. Please go back to slide Software Lab S2 and make sure you understand it. How is GSyn different? GNA was voltage gated. This is a voltage gated sodium channel. GSyn is neurotransmitter gated. It's not voltage gated. That's the difference. And what is the equation for GSyn? As I said, it's I sin is equal to G sin times V minus E sin. And G sin itself is a transient channel. So this channel is open only when the neurotransmitter gates it. So if I go back to this neurotransmitter, so the way it works is when the neurotransmitter is released, it goes and binds to this channel for a short duration only, and then unbinds. When it unbinds, this channel closes. So it's a transient channel. How would you model a transient channel like that? Well, if I plot GSyn versus time, GSyn should increase and then decrease to 0. So meaning this channel increased and then decreased to 0. Okay. So what's an equation for GSyn? Any timed equation which goes up and comes down. So we'll talk about this next. The specific equation you don't need to know, but it's good if you do. So if you want the specific equation, so this is the G max is the G bar. you can write it as t over tau, e to the power 1 minus t over tau. Remember, it has to open and then close. This is G bar or G sin. Okay, it's like this, a rise. So another way of expressing G sin is an equation like this. But if you remember that it has to have a rise and a fall, you can then appreciate it has to have two time constants. One is a rise time constant and one is a fall. And that's what this equation shows, this representation shows. Okay. And then a synapse should also have a reversal potential. So that is typically for AMPA synapses, the reversal potential is 0. And for NMDA synapses also it's 0. For GABA synapses, it's around minus 70 to minus 80 to minus 85. Okay. So the question here was, what is an equation for G-sen? The answer is, the equation is something like this. We won't, hold you down for this. It's good if you remember it. But if you plot it, that equation versus time, this axis needs to be labeled time. An equation, whatever is a curve fit for that, which is some of those are given here. That's an equation. Okay. So individual synapses can be excitatory or inhibitory. Excitatory means when a spike comes down this, if I record from this postsynaptic cell, the voltage will increase of that membrane. Or the soma, if I record it, the voltage will increase because this injected positive ions into this postsynaptic side. If you have an inhibitory input, it will actually draw ions out, positive ions out. So which will depolarize. So that means the voltage will go down. So the voltage up is here, down is here. From resting, wherever the cell was, it'll go down actually. So this is called hyperpolarization of the cell. This is called depolarization of the cell. So if a cell is hyperpolarized, the trigger zone is not triggered, no action potential. While if it's an excitatory input, it's possible the excitation may exceed the threshold at the trigger zone, and this may spike. What happens in real world is there's both excitatory and inhibitory input. If you consider neurons in your brain, a single neuron might receive, like I said, up to 10,000 to 20,000 inputs. It's a huge mess to untangle this excitation, inhibition, and all. If you think about it. But we work perfectly well. When I wake up the next day, my neurons are still firing the same way. So think about it. It's fascinating. Okay. All right. Now, excitatory postsynaptic potential, which I mentioned a little bit, it's graded in magnitude, have no threshold, cause depolarization, summate, and have no refractory period. So if you have very little neurotransmitter released, it'll have small, and then it's possible they may sum many synapses together. So this may be 2 or 3 synapses working together. This may be maybe 10 synapses working together. They can sum, right? So relative amounts of neurotransmitter. If you have a large amount of neurotransmitter, it's possible you can elicit an action potential. Synaptic inhibition, similarly, there are 2 neurons, 1 and 2, that inhibit. So this is the threshold for action potential. So here comes, the cell is resting at this point. First comes an inhibitory synaptic potential. So if it inhibits from, let us say number 1 is inhibitory and number 2 is excitatory. So if the inhibition comes before the excitation, note that this neuron will not spike in this case. So if the excitation comes before inhibition, this could have spiked perhaps. Because remember, this inhibition caused this neuron to go down, the membrane So the excitation has to work very, very hard to bring it up from almost here to here. Now, if this inhibition were not there, the excitation, if they acted from here, it may have spiked. So then your brain uses mechanisms like this. If you want to, let's say, stop this neuron from making this neuron fire, it may put an inhibitory neuron in between and make that inhibit like this so that this neuron will not have make that neuron fire. Okay. Please check if you understand. Sorry, I switched it. This is inhibitory, this is excitatory. If you want this excitatory neuron, number 2, to not excite the cell, you may want to excite this interneuron before this excitation happens. If you excite this interneuron, it will depress the membrane potential. So when the spike from this comes, it will not make the spike. See if you can pause the video here and fully understand what I just said, because that's critical to modeling and understanding what's going on. Okay. So postsynaptic inhibition is via GABA and glycine receptors. They produce something called IPSPs, meaning inhibitory postsynaptic potentials. IPSPs dampen EPSPs, right? This EPSP could have made this alpha. This IPSP dampened it, making it harder to reach threshold. Okay. Presynaptic inhibition, this is the postsynaptic inhibition. Presynaptic inhibition occurs when one neuron synapses onto axon or boton of another neuron, inhibiting release of its neurotransmitter. So that's another presynaptic inhibition, right? Now we go to spatial summation. If you have neurons that, if this neuron is exciting one dendrite, this neuron is exciting another dendrite of the same postsynaptic neuron. If these spikes come together, the total ions injected in the cell will be double. So if, for instance, if only this neuron spikes, it'll create an EPSP only this big. But if this neuron and this neuron spike do it in tandem, like a relay race, this neuron spike raise the membrane potential this much, and then before it comes down, this neuron spikes. And so that may push it about threshold. So this is called temporal summation, because remember, in time, on the time axis, they summed. The effects of two excitatory inputs summed on the time axis, temporal. So without temporal summation, cable properties cause EPSPs to fade because currents will leak out and it'll fade over time and distance. Spatial summation takes place when EPSPs from different synapses occur in postsynaptic cell at the same time, okay, like I mentioned. So temporal summation occurs because EPSPs that occur closely in time can sum before they fade. Again, I would like you to, one way to learn this is sketch this entire thing yourself without looking at this picture. Sketch entire thing and label it too. If you do that, you will definitely don't have to revisit this. You'll be ready for the exam. Those are the techniques to study well. So this is a very nice slide, which I ask on the tests and all, and it's easier for you to answer. This shows spatial and temporal summation. So in this side, let us see, this action potential comes here, and it gives you just one EPSP, the standard. You'll have to plot all this with time, resting potential, -65, VM, and so on, and you're recording at the soma. Now, temporal summation happens when multiple spikes come at the same location, same synapse. So they build, the postsynaptic potential goes up, up, up. And then after the third spike, there is no other spike, then it decays. If there were a fourth spike, it will go up further from here. So this phenomenon is called temporal summation. Spatial summation, can you guess what it should be? as the word says, spatially. So there are 3 synapses that are at different spatial locations. And you have the spike come at the same time on all 3. So then what happens is at the same time, there are 3. One look like this. So all 3, if you add, it'll go up. Right? And this shows a realistic cell with so many positive, excitatory and inhibitory some synapses, more realistic. So you would want to sketch all of this. Don't worry about this. Sketch this, temporal summation, particularly this plot, and spatial summation, because that's something I ask on the exam. So how does temporal and spatial summation work at the ionic level? Provide a step-by-step description at the ionic level to explain the EPSP seen in the soma. All key laws involved in the process should be explained and linked to the description. So you're given this. This happens, a biologist did this recording and got this plot. You're supposed to explain at the ionic level what happens. Let me get you started. So when this spike comes in, it results in the release of neurotransmitters, which go and bind to the postsynaptic side and opens channels there. So this looks like it's EPSP. So this synapse is excitatory. The current will go in then. Positive ions will enter the synapse and cause this EPSP. Now, explain how that EPSP travels from there to the soma. I mean, this is recorded, at the soma. So there will be an EPSP here too, which will be much larger. And this is the EPSP recorded as soma. So you have to explain how these ions entering this postsynaptic cell cause this at the soma. So I'll leave it to you to think about it and explain at the ionic level. So if you have a giant microscope and you focus it on the ions entering here, track those ions. What happens to them is the question. And what are the laws involved as those ions make their way to the soma? What are the laws involved? So what do you see under the microscope? Okay. So try to answer that. And assume this dendrite is passive, meaning there are no sodium or active channels here, only leak channel. Assume only leak channel. Now we come to the lab itself. What are you supposed to do in lab S4? You're given a 3-compartmental model to illustrate the effect of excitatory and inhibitory synapses, right? Note, we have been focusing on a single compartment so far, largely, but now move on to a 3-compartment. The compartments are soma, dendrite 1, and dendrite 2. So we've mixed axon with soma here. That's what it's understood if you do this. So what you're given is, you're given a way you can put in current injection if you want into the soma, or you can put the current injection into the dendrites. That's what these electrodes show. Alternatively, you can put synaptic input into the dendrites. And the synaptic inputs can be excitatory, which is source A, or inhibitory, which is source B. And you can control the frequency of this excitatory and inhibitory inputs, meaning the spikes coming down this channel, this pathway. So you're going to play around and do things with this sort of model, okay? If you wanted, you can model many more compartments and all too. And in research, we do that. My lab does that. We model very realistic. But the ideas of what happens is, this reduced order model is sufficient to explain to you for you to learn what really happens. Okay? So try that lab out, lab S4. That lab S4 has a different part also, a part B, which is earthworm reflexes. So I'll go over this also quickly. I hope you can read it and completely understand. So an earthworm, if you have touched an earthworm, do you know what will happen? If you have an earthworm wriggling around and you touch it, you know what will happen? Can you describe it, please? Write it down. What will happen first? So think about touching it at the anterior end or touching it at the posterior end. So pause this video and write down what you think. And then only continue. Okay. All right. So if you thought about it, what will happen is if you touch the earthworm at the anterior end, it will exhibit something called an escape reflex. It will suddenly shrink back or, move back pretty quickly. And the same happens when you touch the posterior end. It will quickly withdraw, right? And that's the escape reflex. Do you know how that happens? How might that happen? What really happens is when you touch or when you stimulate the posterior end, there are sensors there, sensory neurons. Those sensory neurons spike, and that through an interneuron, that then activates a muscle. So that synapse there between those sensory neurons and the motor neuron, there's a circuit there. That synapse is not very strong. So it takes 3 sensory action potentials, meaning if you stimulate the posterior end, you need to touch it a little harder for it to really exhibit that escape reflex. And I ask you the question, why? Why is that so? If you stimulate the anterior end, just 1 sensory action potential is enough to elicit the escape reflex. Again, the question is why? Why is the difference between stimulating with the same strength? If you stimulate the posterior end, it may not elicit an action potential, while it may for the anterior end. So we will use a computational model to answer this question. Okay. So that's a model given to you, earthworm escape reflex. So why are we studying this? Because in humans, there is a patellar tendon reflex. If you go to the doctor, they just, the doctor does this, hits you on the knee with a slight, taps your knee, and then you see a reflex action here. So what's happening here is, when you hit this, there is a loop through the spinal cord, similar to the same loop that works in an for the earthworm. Okay, that will cause this hamstring muscle to quickly, provide this reflex, in this case, to move forward. Or if you touch a hot stove, what happens is the touch sensory neurons go through your spinal cord. Your brain doesn't even realize you've touched a hot stove. Even before that, your hand jerks back. Think about it. That's a reflex action. So reflexes can be critical for survival. Nature has designed those things in you. You don't even need to be aware of it. For an earthworm, there is a sensory receptor, giant fiber, and that goes through this giant fiber, goes to a motor neuron, and then goes to the muscle. So the sensory receptor, if this fires a spike, if it's at the anterior end, 1 spike here will cause this muscle to move, is the point being made for the earthworm. And we have a lab that you may or may not be able to do, depending on which class you're enrolled in. And sometimes we don't do the lab because it's fairly obvious. We have an earthworm and we show you all this. By touching the anterior end, you see only 1 action potential, and then you see the reflex action. So let me read this. So the giant fiber escape reflex. When touched at either end, a worm will quickly shorten its response, and sometimes flatten near the stimulated end. This completely involuntary response exists to allow the worm to quickly escape from predators. Just like when you touch a hot stove, it's an escape reflex. Anterior escape reflex. So this is an earthworm about, 10 to 12 centimeters long one. Stimulation at the front end activates a medial giant fiber. And these are details if you're interested about the skin. There is a ring muscle and a length muscle. So if you stimulate the sensory neuron in the skin, it will, through this interneurons, it will activate this giant motor neuron. It may activate depending on how strong is the stimulus, and then engage these muscles for the escape reflex. And the posterior escape reflex, this is not very strong. The synapse is not very strong. So you need much more stimulation on the posterior end to make this work. So it's basically showing you how synapses control behavior. Thresholds for reflex activation. For the anterior end, the threshold is very low, meaning a little bit of a poke is enough and so we are measuring the muscle action potential. If you do an extracellular measurement, you can basically get both the sensory action potential and the muscle action potential. And that's what is being shown. In the posterior end, one action potential doesn't elicit a muscle potential. But if you have three of them, then it elicits a muscle potential, meaning the escape reflex is engaged only if there are three sensory potentials, meaning it has to be a strong poke. While in the anterior end, just one action potential is enough to engage to cause the muscle to move. Okay, let me keep moving. So if you do the biological experiment, you'll observe this. Weak stimulus, there is no action, and there is no recording, no action potential. Strong stimulus will give you this, basically it's showing these plots here. Okay. So in the lab, you observed a behavioral response. So with the poke, something happened. Now we ask what neural circuit implemented it, right? So we have this. What we have done is stimulus response. So in the lab, you have a sensory neuron, and we model the LGF or MGF, lateral giant fiber or medial giant fiber, by just a neuron, and then this motor neuron. So if you think about it, when you poke at the anterior end, you don't need temporal summation. One spike is enough to activate the muscle, right? Think about it, or motor neuron. While if you stimulate at the posterior end, 3 spikes are needed. So those 3 spikes could result in temporal summation here. See how the link is between temporal summation and behavior. Right? In synaptic transmission, the importance of temporal summation. If there were no temporal summation, the poor earthworm will not be able to escape from predators which try to hurt it from the posterior end. Okay. Hope you get the idea. And you have this funny worm. One of my students, John Ball, actually made this animation. So John's at NIH now in one of the NIH labs. So he likes all of those. Okay, synapses and temporal summation. I won't go over this. I think I've talked about it enough. This shows a temporal summation with the same idea. And this doesn't get time to sum temporally. These spikes need to come faster. If they come much, much faster, note that it sums very beautiful and how the voltage goes up. This is the voltage axis. Okay. Now in the software experiment, you're trying to basically this is trying to represent this biology. The output of earthworm skin sensory neurons is the spikes that come through this. That's the output. So if one spike comes, the soma should spike. If this is the anterior end. For the posterior end, there are 3 spikes that need to come excitatory for this muscle for the soma or muscle to spike. That's the idea. So you get the big picture then. And in the lab, you will have a source A and a source B for excitatory input. You don't need this current injection input, so don't worry about that. So you'll have a source A, which is excitatory, and source B that is inhibitory. And so attach them and convince yourself, show to yourself, adjust the synaptic weights to show how it could simulate either the poking of the anterior end of the earthworm or posterior end of the earthworm. And in this process, you get some idea about temporal summation. Okay. The big picture in this part was that, you were, for a simple example, using a very simple preparation, you were able to connect from neurons to behavior. This is not possible in humans or rodents. You cannot see the activity of one neuron and relate to behavior because there are thousands and millions of neurons which work together to cause behavior. In this earthworm, you can see just from sensory neurons, a few of them, how they can result in behavior. Okay. And you can also see that with our, this patellar tendon reflex, or when you touch a hot stove, there are lots and lots of neurons in your spinal cord which quickly sense it and then pull your arm back. Okay. Again, just an idea of showing you how you can go from neurons to behavior. And the central concept covered here is synaptic transmission and how to model it. So that concludes this video, and you can proceed to answer questions related to S4 and then come back and view this video because some of the things may not be clear to you. And so you have to do back and forth on this. Okay. So we'll see you next time. Bye.